Thanks, Greg. The one positive thing that I'm taking from the first two discussions is that it looks like the future for cord blood is pretty rosy. I represent a biopharmaceutical company that got into the stem cell cord blood space more than 10 years ago. With a specific interest in identifying opportunities to create discrete therapeutic products for a variety of diseases, hematology, oncology applications, immunologic applications, but also the horizon for regenerative medicine 10 years ago was very murky, yet Celgene at the time, developing an emerging biopharma leadership company, did see this as an important place to invest. So what I'd like to do today is try and give you my perspective on where I see the cord blood banking industry fitting into this very broad area of regenerative medicine. And as Greg pointed out, there are a lot of technologies that are displaced across this smear that's called regenerative medicine. I want to talk to you about the evolution of a service provider, a cord blood banking company that became a product developer in sort of the shadows of a true biotechnology, biopharma company, and how I believe the foundation established in cord blood helps to serve us to build products of a broader nature and potentially broader commercial applications, and where this might all lead. So, broadly speaking, regenerative medicine is using tissues or cells to augment, accelerate, enhance the natural regenerative processes and repair processes of the body, and to potentially even replace damaged or diseased tissues as a consequence of trauma, aging, or other illnesses. Candidly speaking, when I got into this business, and I see some of my colleagues that I've known for more than a decade here, we all kind of assumed that someone would have made the jump to take cord blood from simply a hematopoietic reconstituting product and identified some more mainstream applications for this technology to broaden the appeal to the consumer base we were dealing with, expectant parents. At Celgene, a pharma company, I often hear, since we have a cord blood banking business unit in our company, what have you done for me lately? What new technologies are you developing? What can you enhance in our product offering? So the way I see it is cord blood represents the best. And when I think of cord blood, I think of the broad procurement opportunity that exists when you're recovering the products of a full-term healthy pregnancy. And I see cord blood, in that broad sense, as being the perfect supplier for this emerging field of regenerative medicine. It has cells, it has biomaterials, and most importantly, it has taught us all something important. Everything we've learned through the procurement, the qualifying of donors, and ultimately the handling and ultimately delivery of these products clinically has benefited us, those of us who are trying to take a diversified approach to this field. So, evolving from a service provider to a product developer, Celgene acquired Anthrogenesis back in 2002, and it was really the first large entry of a biopharmaceutical company into this space. And what they did was they enabled us to invest in things I think are important: manufacturing and diversifying the product platform. So, briefly, and this is a little bit of an advertisement about Celgene. Celgene is now a global company. We do about $5 billion in revenue, $30 billion market cap. We have the resources to devote to investigating these opportunities that I'm alluding to. The cell therapy division of Celgene, and I'm going to move quickly through this presentation, we're about a 200-person company inside of a 5,000-person pharmaceutical business. Our company has discrete cell therapy products, that is, products developed like pharmaceuticals to be delivered into specific indications with a specific bioactivity at the heart of the rationale to use the products. Immunomodulation, turning on natural repair processes, or whatnot. We also have an element of our business which takes these cells, along with the biomaterials that we recover at that time we're collecting cord blood, and we put them into constructs we think may have a role in the tissue engineering space, in the surgical device space, et cetera. And then our biobanking business, as Greg coined it, I think represents a nice, stable revenue engine in the back room of our business, which provides access to materials, provides that know-how I mentioned before, and ultimately can help fuel greater adoption of banking as we create greater and greater value to these materials. At Celgene, the cell therapy division is a standalone business. We have our own R&D, we've got our own clinical development organization, regulatory affairs, and so on. So in a lot of ways, you can sort of think that when pharma gets into this business, they've got to be looking at the options to take winners and do with them as they please, spin them out, develop them as wholly owned subsidiaries, et cetera. So establishing Celgene as a global leader in regenerative medicine, the first thing we wanted to do was leverage this cord blood banking legacy. And what I mean by that is taking advantage of everything we learned in recovering those cells at the time of birth. Now, with the early focus of Celgene and Anthrogenesis was to uncover the potential of the placenta as an ideal source of not only hematopoietic progenitor cells, but a broader range of phenotypes that might have applications in the yet to be defined regenerative medicine space. Early on, we felt harnessing the unique biologic potential of the placenta, namely its ability to modify a biologic response, such as immune-related functions, and to stimulate endogenous repair and regenerative processes, would enable us to identify at least some clinical indications to chase. And since that time, we've been able to broaden the offering we can bring to the clinic. As a raw material, I think many of you will appreciate that the leftovers of a full-term healthy pregnancy, pre-qualified during that pregnancy, and then subjected to post-recovery screening technologies such as looking for infectious agents and so on, makes this an ideal raw material for regenerative medicine. Inside of a pharmaceutical corporation, we've been able to deploy the best pharmaceutical development approaches to advancing these technologies from the bench to the bedside. And what this really means is understanding how to, if you identify a discrete cell product that might be useful in immunomodulation, how do you mass produce that? How do you scale it so that you can have a meaningful impact in a large disease? And then how can you formulate it so that it can be delivered and adopted by the clinical community with a high degree of success? It also means that we can work from a position of strength when it comes to having a dialogue with regulators. Now, I'm happy to say that here in the United States, the FDA has been a very receptive agency in terms of understanding the limitations, the challenges that we face, and then working with us to better enable us to meet the standards that are established and also reach to achieve a higher standard for quality, controls, pharmacovigilance, and so on. It also means that a company like Celgene can apply a broad set of research energy to understanding the mechanisms of action of these products, and again, better enable us to identify where they belong. So I want to simply say that as far as I'm concerned, the placenta is a wonderland. It has, as you all know, a variety of different phenotype, progenitor, and stem populations. We were the first to describe back in 2000 that we could recover a pluripotent phenotype from the placenta, and from that we've developed a family of multipotent and pluripotent cells which we are developing for clinical applications. But there are also unique cell types such as natural killer cells, and the leftovers of the placenta after you recover cord blood can be used to augment the potency of that product, cord blood. HPDSC, which is not very creative, a human placental derived stem cells, is simply a composite product recovered by perfusing the closed circuit circulation of the organ after it's been exsanguinated in order to boost the CD34 population that you can offer patients. Real simply, the necessity is the mother of invention. Has been described, cord blood's often been criticized for having a low potency, a low content of hematopoietic progenitors. HPDSC was our approach to solving that problem. In addition, the leftovers of the placenta are an ideal source of biomaterials. The amniotic membrane has already been turned into a product for wound healing, for ophthalmic use. the placenta is rich in collagen. Collagen's a big business in its own right. Placental collagen has many advantages to xenograft material as well as to other forms of human collagen. And then we have recently been very, very good at deconstructing the decellularized placenta, isolating the structural elements and turning those into a product we call ECM, which is nothing more than a matrix material that can be used as a vehicle, a carrier, a delivery system, or basically a raw material for engineering different constructs. And then, believe it or not, and this is something I did as long as 20 years ago when I was a surgeon at Cornell Medical Center. Everyone who's seen a placenta recognizes the vast vascular network on the surface. You literally can dissect these vessels and use them for research purposes and in some cases even clinical applications. So our technology platform is broad. Simply enough, derived from the placenta, we recover a range of cell product candidates seen at the bottom left. PDAC stands for placentally derived adherent cells. That is the first platform from which we developed a clinical candidate. AMDAC is another class of cells which have some unique properties. NK cells, we're developing these for use in cancer. But the nice thing about this is the placenta has turned out to be a very stable source of cells, and the cells we recover are highly expandable in culture, lending themselves well to manufacturing methodologies. I'm going to speak very quickly about HPDSCs because this is the perfusate, if you will, of the placenta, which we first developed to augment the CD34 count you could get from a single cord blood donor. There are major cell populations in this material. Our method of recovery, which is proprietary, derives 100% fetal cells. Unlike some groups that consider themselves placental, but they're deriving cells from the maternal side of the organ, these cells are 100% derived from the fetus. Among the cells in this composite are leukocytes, a variety of progenitors, including committed megakaryocyte progenitors, endothelial progenitor cells, multipotent and even pluripotent phenotypes that can be used in different applications. Our first foray into the clinic with this product was to augment the potency of a cord blood transplant in a young patient with leukemia. And simply enough, this is a N of 1 in this case, but we were successful in boosting the potency of that transplant. Patient was discharged from the hospital 70 days post-transplant and has done quite well. And we consider this to be compelling, but clearly not conclusive. We're looking at taking HPDSC because it can be used as an autologous adjunct to cord blood, but also it's potentially valuable as an allogeneic product into a variety of settings, including augmenting transplantation, potentially as a universal cell donor product, but also autologously in patients who you have the opportunity to recover at the time of birth, and these neonates have an underlying medical condition that requires emergent therapy, perhaps neonatal ischemia, perinatal trauma, etc. There are a number of investigator-initiated trials using this product that are in development, and we're currently developing protocols for sickle cell, potentially using this in the bread and butter of cell gene multiple myeloma, as well as looking at some encephalopathic indications to pursue. We're continuing to develop models, preclinical models, which will enable us to have the right mechanistic rationale to take it into different diseases, but our ultimate goal is that those studies will enable us to move into some of the areas that are listed here. So let me talk about now the discrete product, which is derived from the placenta from this family of cells called placentally derived adherent cells, PDACs. It was the first family characterized from our recovery methods from the placenta. Lots of unique biologic attributes. The ones that are most important to us are the immunomodulatory behavior of these cells. PDA001, the lead clinical candidate, has been taken through phase 2 in a number of diseases, and I'll speak to that briefly. And we're also continuing to explore its potential role as a regenerative effector. One thing that's been very, very important to us at Celgene, and this was a prerequisite for the continued investment by our corporate parent, and that was to demonstrate that we can produce products to the scale that they are expecting of a biologic. That means that from a single placenta, we obviously couldn't have that single donor, single recipient relationship. We had to broaden this, so from a single donor, we could mass produce. And I'm happy to say that in our hands, we've reached scales that we believe, we're confident, will enable us to produce hundreds of thousands to perhaps 1 million doses per placenta. That's the type of scale that is commercially relevant to a pharma company. Our focus, immune modulation and supporting the natural regenerative processes in the body. We've reported some of our data in Crohn's disease, multiple sclerosis, and we've got news coming on sarcoidosis. And I'm happy to tell you that we're excited enough about these things to continue advancing this product further into further phase 2 and phase 3 studies. But all of this, because Greg, I think, alluded to the fact that the business model that seems to work is diversification. Right? And the way I look at it, if you're going to invest a lot of energy in recovering the leftovers of birth, why not recover everything you can, spread the cost of procurement, qualification, quality control across several product platforms? And so there are obvious synergies and competitive advantages to adding tissue engineered constructs to what you could get out of this procurement event. So the placental technology I alluded to is obviously important because of the cells, but it's also important because of the biomaterials. And if you actually combine these, the economic modeling becomes far more attractive. So from the placenta, I spoke about the cells we recover. Here are some of the materials we recover. The amniotic membrane, many of you may know, has already been developed by Anthrogenesis and now Celgene into a native tissue matrix that is decellularized, sterilized, and dehydrated, so it has a long shelf life. And this natural tissue matrix has had tremendous success in everything from ophthalmic surgery to burns and to other skin injuries. Placental collagen is a product that continues to intrigue us because of the obvious value to the cosmetic and aesthetic surgery markets. And then ECM, chorionic matrix and extracellular matrix, as well as potentially Wharton's jelly or cord or umbilical cord matrix, we feel is a rich opportunity to create many building blocks for engineered tissue products. Very, very briefly, this isn't rocket science, but after you get all the cells out of the organ, we were the first to describe an economical and a reliable method to remove all the cellular immunogenic properties of the material, and then break it down into its finite structural elements, and then reassemble those finite structural elements in useful forms. And what you see here is just like if you demolish a house, if you do demolition, demolish a house, and then recover the bricks and use them to build another house, that's sort of what we're doing here. And right now, this product is in clinical application, in reconstructive uses in post-traumatic injuries. We've got a relationship with the Department of Defense in this space, and we think that this will also be an important delivery system for delivering cells to those types of indications. ECM is great because as a raw building block, it can be put into a variety of delivery systems. It can be injected as an emulsion or a suspension. It can be assembled with or without other components into different useful forms, and it lends itself very well to the surgical environment. I can tell you as a surgeon that if I had this stuff on the shelves when I was taking care of trauma patients 15, 20 years ago, I would have used it in every case, because there's always an opportunity to fix a defect with a living native biologic product. This wound care product is sterilized. The nice thing about it is, as you break it down to its finite structural elements, you can use sterilization techniques which don't alter that native biochemical architecture, and so behavior when transplanted and used in patients is quite normal, and it can be packaged in easy, long shelf life forms. The long story here, not the short story, but the long story is that ECM as a platform, in combination with cells, in combination with small molecules, peptides, other chemicals, lends itself to many of the long view opportunities that exist in tissue engineering. Can we build an organ on this platform? We don't know. It's possible that somebody figures out a lot of the assembly technology. But can we build an organoid? Can we build a discrete material that can deliver phenotypically specialized cell types? Yes, we can. We've already begun to take ECM, break it down into its finite material, and then use it in essence as the ink in a three-dimensional bioprinter. And what we can do is we can create constructs using bioprinting, such as this small wafer that we can load with cells, differentiate into discrete specialized phenotypes, and we call them finite physiologic functional units. I can grow 1 million hepatocytes in this material. It's completely biocompatible, and this can be delivered to a patient subcutaneously, intraperitoneally, or it can be loaded into an extracorporeal system. So this, we think, holds a lot of promise for taking all these materials recovered at the time of birth and putting them to good use. So in summary, because I know time is short, we believe that cell therapies will always be a pillar of this business, but we also believe that the organ and tissue engineered applications will be a buttressing pillar that will enable us to derive higher returns for our investors, and more importantly, will provide us constructs that may be able to demonstrate clinical efficacy of the cells before discrete cell products might even reach that level of approval. The combination product opportunities are obvious and vast, and we do believe that the industry is going to see greater participation, larger companies that will probably see the tissue engineering applications combined with cells as risk mitigating, and again, the investment opportunity then becomes a much more attractive. And where is this going to lead? Well, I think that regenerative medicine as a field is going to require tremendous collaboration. Some of the work that I've heard this morning tells me that as cord blood becomes ever increasingly important in clinical medicine, this source of material will have greater credibility. It's validated. There's comfort in the clinical environment, and that cord blood got me here. If we weren't a cord blood banking company, we wouldn't have embarked on all this other discovery, which led us to where we are today. And I think that Celgene Cellular Therapeutics, as a company within a larger pharma company, represents an important model for other biopharmaceutical company constructs. Thank you very much.
All right, we're short on time, but there is one question I need to ask. How do you wrap intellectual property around the cell therapy components of your business model? Lots of folks are working with cord blood, placental tissue. Can you describe just briefly your intellectual property strategy?
Sure. So when it comes to the phenotypes we recover from the placenta, fortunately, we were a little bit ahead of the curve, and we laid a strong foundation in placentally derived progenitor cells. However, as I mentioned at the end of the talk, I do believe collaboration in our field is essential. Mm-hmm. So as a construct, for example, using the biomaterials, might rely on cord blood elements, I think we're going to have to create the kinds of joint ventures and business relationships which enable us to bring this to the market without conflict, with a focus on sort of sharing the wealth, mm-hmm. And I think that unless you do that, you're going to wind up having the types of long-standing unharmonious relationships like our friends who litigated over the original cord blood
right.
patents.
Yes, so. I couldn't agree more. Okay, any other questions? Quick question? No? Okay.